Okay, good evening, everybody. It is my great honor to have a colleague from Loyola University Medical Center in Chicago, USA, and to share us our weekly scientific activity in Acod. And it is a great honor to have Dr. Loyola biosimilars, a topic which I believe is very crucial, not only to developing countries and poor countries, but also for rich countries. Currently, the medication for oncology is quite expensive, and it is costs to related to medication of anti-cancer drugs. I have to thank my dear colleague Tamer Rifat for giving us opportunity to have Dr. Hannah Mai with us today. And Dr. Hannah, actually, we are broadcasting along Africa, as many of our fellows are in Kenya, Tanzania, Nigeria, Rwanda, and many other African countries. Please welcome Dr. Mai with me, and we are so eager to hear from her. Dr. Mai, please.
Oh, okay. Thank you so much for the invitation. It's really an honor to talk with you about a topic that really got me started into thinking back in 2018, 2019, along with a colleague, Dr. Kitani. I do not major in economics. I don't have an MBA, but certainly it's something that we all need to be aware of because healthcare is expensive. And so that leads to my thoughts and maybe a research into this topic that pertains to everyone, not just developed countries, but also developing countries as well. So my presentation to you today is going to be on next generation biologics in oncology. And I don't have any disclosures, so I don't have any funding or anything. I'm going to start off the presentation with two questions, and I want you to keep in mind which answer you would pick, okay? So the first one is, which of the following is considered a biosimilar? Filgrastim, TBO filgrastim, PEG filgrastim, or none of the above? The next one is a true or false. I want you to keep this in mind, and I hope to be able to help you get an answer at the end of the presentation. So a biosimilar can be used for all indications where a biologic agent is approved. US FDA requires biosimilars to go through the same development and regulatory process as generic medications. US FDA requires biosimilars to go through the same development and regulatory process as the original reference product. There are currently 5 oncology biosimilars approved in the US market: filgrastim biosimilar, pegfilgrastim, trastuzumab, bevacizumab, or rituximab. So keep your answers in mind. These are my objectives for today within the next 40-50 minutes to really understand the definition of a biosimilar agent, understand the regulatory approval for biosimilar agents, mostly in the United States where I practice. Recognize the US FDA approved biosimilar agents. So these are the classes of medications. And I'm just going to provide you with some details of the cost analysis and the economic implication of substituting a biosimilar, and some concluding points and future perspectives, and then my references at the end. So, introduction. Biologic agents have been used increasingly in the treatment of a variety of diseases, not just cancer, so your rheumatologic and other disease entities. Within oncology, however, the market, biologic agents account for more than half of that. So we use that a whole lot in oncology. Patents for the first generation biologic agents have either expired or are about to expire, therefore opening the market for alternatives, agents such as generics and biosimilars. So really, as healthcare professionals, we really need to understand what it means to be a biosimilar and how it can inform our treatment decision. There have been guidelines to educate us, and also there are regulatory pathways to bringing these agents into the market, namely the European Medicines Agency, and within the United States, we have the Food and Drug Administration. I'll refer to that as now the FDA. So this is within the United States, okay? So in 1902, Congress passed the Biologics Act. In 1930, the hygienic laboratory becomes what we know now as the National Institutes of Health, or the NIH. The Division of Biologics Control was formed within the NIH in 1937. And between the 1950s through the 70s, that's really, we experienced tremendous progress in biologic agent regulation. And in 1986, we have the first recombinant DNA vaccine, and it got cut off, but in 2015 was when the United States actually approved its first biosimilar agent. So what are biosimilars? Biosimilars are unique molecules and are not generic versions where the active substance of a generic is identical to the reference product. Really, this serves as a basis for biosimilar approval guidelines. They're defined as biological products similar but not identical to the reference products that are submitted for separate marketing approval following expiration of the patent. Regulatory guidelines really must account for the differences between biosimilars and their reference products. The goal of developing a biosimilar agent is really to create a price competition to provide cost-effective alternatives to biologic agents and really ultimately improve patient access and patient outcome. There are complexities in production, evaluation, regulation, and its clinical application. What are some of the benefits of biosimilars? I had mentioned cost, right? And we had mentioned that the biologics are losing patent protection soon. Therefore, biosimilars have a way now to significantly improve access to patients. FDA-approved indications for biosimilars do not require a clinical trial for each indication, thereby improving costs as well as time. And FDA approval process allows expedited development of biosimilars and rapid entry into the world or into the market and improving access altogether. So this is a very nice slide showing differences between biologic agents and small molecules. So I will refer to kind of the left-hand side, okay? So small molecule drugs generally are low molecular weight, whereas biological products are high molecular weight. The small molecule drugs are made by organic or chemical synthesis, whereas biological products are made from live cells or organisms. There's many critical steps. There may be fewer critical steps in the small molecule drug development. They tend to have homogeneous drug substance in the small molecule drug, in contrast to a heterogeneous mixture in the biological product. Often not immunogenic in small molecule drugs, whereas often immunogenic in the biological products. And to the right-hand side here, I'm thinking about, small molecule drug, you're thinking of an aspirin, right? And then thinking about a large biologic agent, you're thinking of the IgG antibody. Analogy wise, think about small molecule drug as a bicycle versus an airplane, so to speak, with a biologic agent. Biologics and biosimilars, okay? So biologics, they're large and they're usually complex molecules made from living organisms. And they include a wide range of products such as vaccines, gene therapy, blood and blood components, somatic cells, recombinant therapeutic proteins. Biosimilars are products highly similar to an FDA-approved biological product. They have no clinically meaningful differences in terms of safety, purity, potency, and effectiveness from the reference product. And up top here, you can see they're almost identical looking, except for maybe a minor that's not clinically significant. Oops. So how does a biosimilar, how is one developed? And so usually starts with reverse engineering to create a biosimilar from a reference product already. And this product goes into in vitro studies, so lots of physical, chemical, and biological studies to characterize it. And these studies will need to determine safety. And then these prelim data are presented to see if this particular agent can progress to clinical development. If the answer is yes, and there's more comparative pharmacokinetic, pharmacodynamic, and immunogenicity studies. And then this is done in a sensitive patient population. Again, the data will be presented to regulators. And again, when regulators assess this, they assess the totality of the evidence from in vitro, in vivo, and clinical studies to see if it is or to see if bioequivalence is established. That will lead to its approval. So the regulatory process for biosimilar is not the same as that used for small molecule generics. Regulatory agencies have established guidelines with minor differences. So usually these involve a stepwise approach that includes analytical studies, at least one pharmacokinetic study. There's a minimum of one efficacy and safety intended to support a demonstration of biosimilarity. And really, ultimately, as I mentioned before, the approval is based on the totality of the evidence in support of the biosimilar in comparison to the reference product. Okay, so here's a nice slide from a review article. So these are two. So these are biosimilars, proving highly similar to the reference products via all of these. So you see that this can be considered as a pyramid. In the base of the pyramid, you're relying on physical, chemical studies, biological studies, and preclinical, and pharmacokinetics, pharmacodynamics, and then clinical trials, leading to the approval of these. This is something that the FDA actually has on their website in the United States to approve biosimilar. So really establish biosimilarity based upon totality of evidence, not reestablish benefit using this abbreviated development program, according to our US FDA. I just put this in just to kind of remind ourselves that, manufacturing biologic agents, not only is it expensive, but it's time consuming. It involves a lot of processes, right? Cloning, protein expression, and purification and validation. So this could take years. What are some of the requirements that are coming from the US FDA to be able to approve a biosimilar? So FDA guidelines include a biosimilar utilizes the same mechanism of action for the proposed conditions of use, but only to the extent of the mechanisms are known for the reference product. The conditions of use proposed in labeling have been previously approved for the reference product and has the same route of administration, dosage form, strength as the reference product, and is manufactured, processed, packed, or held in a facility that meets standards designed to assure that the biological product continues to be safe, pure, and potent. Naming. The FDA does have guidance on naming the biosimilar. So for example, this is to really facilitate pharmacovigilance and to prevent inadvertent substitution. So in the United States, a biosimilar is named using the international non-proprietary name plus a random 4-letter suffix for all biologics. So these are just some of the examples. So filgrastim, SNDZ, trastuzumab, DKST, etc. In the United States, and going back to just 2008, but between 98 to 2008, you can see the market for the small molecule use in biologics. And you can see that about a third in the market are biologic agents. So we're using that. And I can only imagine if we were to sample kind of, from 2009 till now, this number continues to be increasing with time. And this is a nice slide to compare the cost and the time that it requires to make a biologic agent versus a biosimilar versus a small molecule generic. Okay, so here, according to this study, it takes 1 to 2 million over the course of 1 to 3 years versus biosimilars 10 to 40 million, 6 to 9 years versus a new biological agent, billions and billions of dollars in 10 plus years. This is from a recent study. Again, same thing. It's adding generic drugs. Again, just kind of illustrating that, it takes longer and more expensive to really make the biological drugs, lesser with similar drug biosimilars. Of course, the least would be the generic. So this is the slide that kind of got me into thinking why we just don't use this earlier on, but this was reported in 2014 and really saying that the specialty medications in the United States keeps on going up, and it's been going up 4 times since 2006. Okay, and this includes biologic agents. If we were to approve biosimilar agents, as mentioned here, the United States could be saving billions and billions of dollars, according to this article here. We know that biosimilars are safe because our European colleagues have been doing it since 2006, 2007. Japan's been doing it since 2009. Canada and Australia since 2010. Actually, more recently, I read a paper where Australia is actually investing 20 million just to have their own healthcare providers and stakeholders know more about biosimilar and its regulation, and to think more about implementing biosimilars. Korea in 2012 and 2013, India, and in the United States, the first biosimilar was approved in 2015. And this is why they're less expensive, again, not to the same degree, right? So here, according to this study, the difference, the discount in generic is 75% versus only maybe 10 to 30% using a biosimilar. Still, it's still a difference. So these are the US FDA approved biosimilars. Okay, this is filgrastim, pegfilgrastim, trastuzumab, bevacizumab, and rituximab. Okay, and these are their biosimilars, and you can tell that the biosimilars have the 4-letter suffix here towards the end. And the first biosimilar was approved in 2015 was the filgrastim SNDZ from Sandoz. And just a slide on the clinical trials and biosimilars, kind of the design and what they look at and the statistical really design of that. So really the goal of a comparative clinical trial is to demonstrate equivalence of the biosimilar and the reference product. So in a lot of these trials, you'll see mentioning the equivalence design, okay, where the difference in efficacy or safety between the biosimilar and the reference product is less than a pre-specified margin of clinical equivalence. And the equivalence is usually assessed using 2-sided confidence interval for the difference between treatments, typically 90% or 95% level. Survival endpoints may not be appropriate because of potential confounding due to disease status, previous lines of therapy, but response rates, progression-free, pathologic complete remission or response may be appropriate. And you'll see that as we have examples throughout the presentation. So in 2015, there's about 14 biosimilars approved in the United States, okay? About 99 of those are oncology and oncology supportive care medications. And these, to list a few here. And this is from a recent review article that was published last year, indicating kind of all of the biosimilars that have been approved in the United States, in oncology in particular, and their FDA approval date here. So I mentioned 2015 was Zarzio or SNDZ filgrastim, and then most recently Rituxan from Pfizer was approved. And this is same review article, but this is from Europe. So Europe has been ahead of us and has been approving or using biosimilar at least in 2007. And these are all of their approval agents to date. So I'm going to talk about filgrastim. This is our first US approved biosimilar. As you know, this is a growth factor. So filgrastim SNDZ has been approved as a biosimilar drug, not an interchangeable drug. Those are different designations. And at the time of the approval, it was priced at a 15% lower cost than the reference drug filgrastim. And I kind of gave you a few numbers in terms of the cost reduction here, but about 15%. So the FDA looked at it, and when filgrastim SNDZ went in front of the FDA, they were able to prove all of these, demonstrate high similarity using the provided analytical data, animal studies, clinical studies, and prove that it's the same as the reference drug in terms of mechanism of action, route of administration, dosage form, and strength. And these are all their fulfillment of the requirements. So really they get all checked, boxes checked here. So this is the clinical trial that led to its approval. So this was studied in 218 breast cancer patients undergoing myelosuppressive chemotherapy. This was a 1-to-1 randomization. And these are patients getting neoadjuvant or adjuvant TAC. So their primary endpoint was mean duration of severe neutropenia, and their pre-specified equivalence was within 1 day. And you can see the Zarzio and the Neupogen. So this is a biosimilar. This is the Neupogen. 1.17 days, 1.2 days, and their difference of 0.04 days, which is well within the pre-specified equivalence range here. So therefore meeting criteria or meeting statistical significance. They also provided secondary endpoints here, and you can tell not a whole lot different. And this is their depth of ANC nadir in cycle 1. And you can see that the curves are almost superimposable. Similar side effects. And overall, again, similar overall safety profile across all 6 cycles with continuous treatment. So that led to its approval in 2015. This study, the PROTECT trial, looked at the use of Neulasta or pegfilgrastim in also breast cancer. This is a prospective randomized double-blind multi-center confirmatory efficacy and safety study. This was a head-to-head comparison between these pegfilgrastim biosimilar, this is the name of it, with our reference pegfilgrastim. And the study was designed to show equivalence in the reduction of duration of severe neutropenia in breast cancer patients receiving myelosuppressive chemotherapy. They stratified by chemotherapy, adjuvant or neoadjuvant, 1-to-1 randomized. The primary efficacy endpoint, similar to the other trial that we mentioned, was mean duration of severe neutropenia during cycle 1. And these are some of their secondary endpoints: depth of ANC nadir, time to ANC, etc. And their statistical analysis. 90% power for testing of equivalence at the 2.5% significant level, assuming there's no difference in the mean duration of severe neutropenia between treatments, with a common standard deviation of 1.6 days. And they establish equivalence for difference if the confidence intervals were within plus or minus 1 day. And let's see if they achieve that. Okay, so this is the full analysis set, and this is per protocol cohort here. And if you look at mean and median, and you look at the treatment difference, 0.07 and then 0.04, again, within that 1 day of the pre-specified equivalence. This is more secondary efficacy parameters and similar in the 2. And then these are the curves here of mean ANC, and you can see they're pretty much equivalent. This is incidence of serious neutropenic events. And so the conclusions of the PROTECT-1 trial said pegfilgrastim biosimilar was equivalent to the reference pegfilgrastim in terms of efficacy and safety in the prevention of neutropenia in patients with breast cancer receiving chemotherapy. That was in the United States. And then this one is actually comparing the US license and Europe license reference product to the biosimilar here, also in breast cancer patients receiving myelosuppressive chemotherapy. So this trial here was a randomized, controlled, blinded, multi-center, comparing efficacy and safety of pegfilgrastim biosimilar to the US pegfilgrastim and Europe pegfilgrastim reference products. And this study included 11 Central and Eastern European countries. The randomization occurs in a 2:1 fashion, and they define their equivalence was minus 0.5 to 0.5 days. If the difference falls into this, then that would mean that they're equal or there's bioequivalence. So what did they find in this study? They found that the primary endpoint, which is the duration of severe neutropenia, was comparable across all three treatments. So the biosimilar and the US and the Europe pegfilgrastim reference products. Moreover, the secondary endpoints, which included depth and peak of ANC nadir, time to ANC recovery, post-nadir, and rate of febrile neutropenia, also show similarity among the three groups. So this is just a summary of the duration of severe neutropenia in cycle 1. And these are the median days and median. And if you look at this, and this is the difference here, kind of within the range. So really this head-to-head, this is what they concluded. This head-to-head randomized clinical confirmatory study of a proposed pegfilgrastim biosimilar demonstrates similarity to both the US and the European reference product with regard to safety and efficacy in the treatment of chemotherapy-induced neutropenia in breast cancer patients receiving myelosuppressive chemotherapy. Right. There was a choice in one of the questions about TBO-filgrastim, and I don't know if you guys use this a whole lot, but we certainly do. But this is a recombinant human granulocyte colony stimulating factor. FDA approved to reduce the duration of severe neutropenia in adults with non-myeloid malignancies receiving myelosuppressive chemotherapy with significant evidence of febrile neutropenia. It's not technically considered a biosimilar because it was filed as a biologics license applications since the biosimilars approval pathway had not been established at the time of FDA submission. So TBO-filgrastim and filgrastim have slight structural differences. However, the pharmacokinetic parameters, safety and efficacy between the two do not significantly differ. But technically, TBO-filgrastim doesn't fall under the biosimilar umbrella. There have been 3 key clinical trials published to date to establish that TBO-filgrastim is an equivalent alternative in terms of efficacy and safety. All right, Avastin or bevacizumab. I certainly don't use this drug a whole lot since I don't do GI oncology or neuro-oncology, but I think we all know that it's a monoclonal antibody directed against VEGF. It's been approved in the United States and in Europe for a wide range of cancers, lung, cervical, we mentioned GI cancers. There's been, multiple randomized phase 3 trials that have shown an advantage, both PFS and OS in patients with these type of cancers where Avastin is added to chemotherapy. But patient access to Avastin may be limited due to cost, right? That could be a barrier. And in some areas of the world, the patency has expired, some in 2010, some soon, some have already. There was a phase 3 trial looking at patients with non-squamous, non-small cell lung cancer, comparing the Avastin and this ABP 215, which is an Avastin or bevacizumab biosimilar. And it did show that the objective response rate was within the pre-specified margin, therefore demonstrating clinical equivalence. In addition, safety and immunogenicity were also comparable of the two between Avastin and its biosimilar, ABP 215. So I included this because these are clinical studies in lung cancer looking at the different Avastin or bevacizumab biosimilars here from Pfizer, from Amgen, for example. Trastuzumab, something that I'm more comfortable with because I also treat patients with breast cancer, but we all know that it's a monoclonal antibody that binds to the extracellular domain of HER2, blocking receptor activation and proliferation of cells expressing HER2. It induces the downstream effects of antibody-dependent cellular cytotoxicity in and cellular phagocytosis of HER2-expressing cells. It's approved for the treatment of early stage and metastatic breast cancer, as well as metastatic HER2-positive gastric cancer. And in the United States, actually, the patency expired in June of 2019, and in Europe earlier than that, so July of 2014. So I included these phase 3 trials of trastuzumab biosimilars. You can see that this is in South Korea. This is one. This is another product from Pfizer in USA here. This is one from Amgen in California, and then the rest here. And its approval status on the right side. And I'm just going to bring about this, the LILAC trial. I selected this. So this looks at the efficacy and safety of one of these trastuzumab biosimilar, and this was from Amgen, compared with the reference trastuzumab in patients with HER2 positive early breast cancer. And this was a randomized double-blind phase 3 trial. This is a controlled equivalence trial, and it was at 97 study centers in 20 countries, mainly in Europe and South America. And after 4 cycles of anthracycline-based chemotherapy, patients were assigned to receive 1-to-1, so either the biosimilar ABP 980 or the trastuzumab with a permuted block design, so blocks of 4, and it's computer-generated randomization. Their co-primary efficacy endpoints were risk difference and risk ratio of pathological complete response in both breast tissue and axillary lymph nodes, assessed at a local laboratory, but also at a central laboratory. And so this is what they reported, and this is PCR, and this blue here is the ABP, so the biosimilar, and this is the reference product. So A here, based on local laboratory review, the risk difference exceeded the predefined equivalence margins. And here, from a central laboratory evaluation, they showed similar efficacy of the 2 drugs, with both risk estimates contained within the predefined equivalence margins. This is, again, looking at the side effects, so the infusional reactions, neutropenia, infection, hypersensitivity, cardiac and pulmonary toxicity, varying grades in the biosimilar and the trastuzumab, and then once they switch, recording their toxicities there. Pretty similar, same thing here. Again, same thing here. Okay, so what did they conclude? They concluded that the response rate of pathologic complete response and risk difference showed non-inferiority. Estimates for the 2 drugs were contained within the predefined equivalence margins, indicating similar efficacy. The ABP 980 biosimilar and trastuzumab had similar safety outcomes in both the neoadjuvant and adjuvant phases of the study, and safety and immunogenicity were similar in patients in the 2 groups. Lastly, I wanted to bring up, because this is actually fairly recent, and we know that it was approved in 2019. This was published in 2020, and this is a randomized, double-blind efficacy and safety study of this rituximab biosimilar, this long name here, compared with rituximab reference product in patients with previously untreated CD20 positive low tumor burden follicular lymphoma. And this was reported by Sharman. And this is their trial schema here. Patients were randomized either to the biosimilar or the Rituxan, so 1-to-1 randomization, and they were treated weekly, days 1, 8, 15, and 22. And these patients were follow-up, and primary endpoint at 26 weeks were generated, and then at the study completion, which is at week 52. And this is what they found. So the primary endpoint was overall response rate at weeks 26. This is the biosimilar, this is the Rituxan reference. And they achieved the pre-specified equivalence margin. And this is indicated here. So this is what they generated. So the results of this study showed that there's therapeutic equivalence between the rituximab biosimilar and the reference product at week 26 for the primary endpoint. Based on central review assessment, there was no clinically meaningful or statistically significant differences in the CR rates or response rates at week 26. And all of the other things that they also looked at, PFS, for example, duration of response, etc., all of which were similar between the Rituxan and Rituxan biosimilar. Both showed similar safety profiles, and the immunogenicity and the pharmacokinetics and pharmacodynamics were also similar during the study. So what we should know about biosimilars, as we see patients and prescribe treatment. So I think that we should know what a biosimilar is, right? We should know regulatory approval information within your area of practice. Know what are some of the approved indications, the status. Having interchangeable status is different. I mean, that has a different kind of sets of criteria that you have to meet. Know your substitution practice within your country or your state. I mean, I live in Illinois. That might differ if I were to live in California. And know the approved indications. NCCN, so the National Comprehensive Cancer Network, and this is what they their stand is on their biosimilar. Biosimilar clinical trials should include endpoints as response rates, progression-free survival, etc. The FDA should be consistent and transparent in how it evaluates biosimilarity. The individual pharmacy and therapeutic committees should evaluate biosimilars considering their institution's specific patient population. And the guideline panel should evaluate and provide recommendations on the clinical use of biosimilars. And so really, we use NCCN guidelines a whole lot in terms of staging and treatment of our patients. And it seems to me that the NCCN guidelines is offering kind of information for us to know, and they recognize the importance of biosimilars and how useful it can be. What about our American Society of Clinical Oncology? What do they have to say about biosimilars? And so they really offer clinicians guidance on the use of biosimilars. There's articles on safety and efficacy. There's articles on definitions of interchangeability, switching, and substitution. There's education on naming and labeling. And also there's information for the prescriber and as well as for patients to know. So these statements really, I think, affirm the society's ongoing commitment to enhancing confidence in biosimilars through provider education advocacy for federal and state policies that ensure really approval, unrestricted access, and appropriate use of biosimilars. So in conclusion, biologics are used widely in the treatment of cancer, but also the supportive care of cancer. Biosimilar products are those that are similar to the reference product, notwithstanding minor differences in clinically inactive components, and that there are no clinically meaningful differences between the biological product and the reference product in terms of safety, purity, and potency. Biosimilars are not generic versions of the reference products. The regulatory pathway for biosimilars, requires, really the totality of evidence that includes a comprehensive analytical assessment and a clinical comparability study in a relevant disease patient population. We looked at a number of approved biosimilars in the United States. We talked about 4 classes, and I kind of went over, although very brief, their clinical trials that led to their approval. Biosimilars offer the healthcare system an opportunity to reduce cost. I think I mentioned that, 10 to 20%, even 30% in some analyses, and increase patient access to potentially life-saving treatments. So physician awareness of the potential differences between biosimilars and the reference product, as well as the impact on safety and efficacy, is really critical, right, for patient safety. and there's rigorous regulatory framework, and there's also post-marketing surveillance. I think that's important to ensure that safety and efficacy are captured in the real-world setting. And in general, I think practitioners need comprehensive information on biosimilars to make informed treatment decisions, both for their patients and then also for the institution. So going back to our question, I hope that, which of the following is considered biosimilar? I hope that you picked none of the above. I purposely did this because, if I had put filgrastim and then I added the 4-letter suffix, then I think you would have gotten that. But yes, none of the above, because the naming is not right here. And biosimilar can be used for all indications where biologic is approved. Yes, that's true. The FDA, the US FDA, requires biosimilars to go through the same development and regulatory process as generic medications. No, that is not that's not right. FDA requires biosimilars to go through the same development and regulatory process as the original reference product. No, that's not right either. There are 5 oncology biosimilars approved, or classes, I should say, in the United States, and that is right. So Avastin, Rituxan, the pegfilgrastim, filgrastim, and Avastin or trastuzumab. So that's right. So these are my references, and that's it.
Thank you, Hannah, for this very illustrative presentation, and I am glad that you share with us this important presentation. And I think Dr. Abbas have questions. I don't know, ICOM, can we have the audience sharing with us to say this, to share in the discussion, or I have to read the questions for Dr. Mai?
No, I actually can see the question. So
Okay.
I see the first one is from Dr. Goali.
Yes.
Yes, I'm sorry. The question is, different form of the same drug, a subcutaneous form for Herceptin needs a patency period, or it can be biosimilar manufactured without expired patency like that's a good question. But normally, in order to make its way into the market, its patent has to expire first, and then you within the US a new application. And you can do it as a biosimilar application, and that goes through kind of an abbreviated regulatory pathway. But I'm pretty sure that it has to expire first, and then you have to apply through whichever pathway that you want to apply for, whether it be a biosimilar or whether it be kind of this a re-extension of that.
Okay.
Can we use biosimilars? And this is Dr. Abbas. Dr. Omar, can we use biosimilars in other indications rather than indications using comparative studies? No, as of right now, as far as I know with the FDA, it has to be the same indications as the reference. And, they have to kind of, the dosage and the route of administration, all of that have to be the same.
We have any other questions? All right. I think by this, we are really appreciating having you with us today, and we are looking forward that we may have another meeting with you, inshallah, soon. And I would like to thank all the attendees, and I really appreciate the attendance of our colleague by the end of a very long day today. Thank you so much.
Well, yes, thank you for the invitation, and thank you for listening to me to talk about this presentation. And I'm sorry, I didn't realize it's 8 p.m. there. So thank you. Thank you for your listening.
Thank you. Bye-bye.
Take care. Bye.
Bye.